Now we are discussing about glycolysis. Glyco means glucose, lysis means breakdown. The breaking down of glucose is known as glycolysis. The ultimate goal of glycolysis is one molecule of 6 carbon glucose break down into two molecules of 3 carbon pyruvate while producing ATP and NADH. In simple words, one molecule of glucose gives rise to two molecules of pyruvate. But remember one thing, glucose does not turn into pyruvate in a single step. It happens through a 10-step process. For better understanding, these steps are divided into two types. One is invest phase, it means we use ATP, and another one is payoff phase, it means we produce ATP. And first to fifth steps are invest phases and 5 to 10 steps are payoff phases. This all process occurs in cytoplasm. Let us see how it will be happens. Glucose converted into glucose 6-phosphate by an enzyme exokinase. Glucose initially uncharged molecules, so it easily cross the membrane and entering into the cell. Then after phosphate will be added. This phosphate donated by ATP and added by kinase enzyme. If we never added the phosphate group, it become uncharged molecule, so it again cross the membrane. As a result, glycolysis will be inhibited. That is the reason phosphate will be added. Glucose 6-phosphate converted into fructose 6-phosphate via an enzyme phosphoglucoisomerase. These both are isomers. There is no change in the phosphate number, there is no change in the carbon number. Here, fructose 6-phosphate converted into fructose 1,6-bisphosphate. Here, one more phosphate will be added. This phosphate donated by ATP and added by the phosphofructokinase enzyme. Fructose 1,6-bisphosphate cleaved into DHAP and G3P via an enzyme aldolase. Fructose 1,6-bisphosphate initially unstable molecule, so it cleaved into two molecules. One is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. DHAP and G3P are isomers. Fructose 1,6-bisphosphate contains 6 carbons and 2 phosphates. These are cleaved into 3 component of DHAP and 3 component of G3P. These 2 phosphates also shared into DHAP 1 phosphate and G3P 1 phosphate. DHAP and G3P both are isomers. Whatever the function performed by one component, the same function performed by another compound. That is the reason we consider one component while calculating DHAP and G3P. G3P converted into 1,3-bisphosphoglycerate via an enzyme dehydrogenase enzyme. Here one more phosphate will be added. This phosphate donated by cytoplasm. 1,3-bisphosphoglycerate converted into 3-phosphoglycerate via an enzyme phosphoglycerate kinase. Here losing of one phosphate, that phosphate accepted by ADP. 3-phosphoglycerate converted into 2-phosphoglycerate via an enzyme phosphoglycerate mutase. Here there is no changing in the carbon number, there is no changing in the phosphate number, but phosphate shifted from third carbon to the second carbon. 2-phosphoglycerate converted into phosphoenol pyruvate via mutase enzyme. Here also there is no change in the carbon number and phosphate number, but releasing of one water molecule. Phosphoenol pyruvate converted into pyruvate via pyruvate kinase enzyme. Here losing of one more phosphate group, that phosphate accepted by ADP.